After studying this module, you shall be able to know about collection of biological evidences, about packaging and documentation of evidences, and about the storage of evidences. Now, we will be starting with the introduction. Blood, semen, saliva, sperm, hair, bones, teeth, tissues and other body fluids which are recovered from the crime scene are collectively termed as the biological evidences. Various types of biological evidences are found at the crime. Every evidence has its own importance and its own probative value. These evidences are mainly used for DNA investigation. Before collection, these evidences are photographed at the crime scene and then sent to laboratory after collection. DNA specific to each individual serves as an effective means of introduction. The various materials which should be considered as a source of biological material are clothing, mask, hats, gloves, etc. from any involved individual, ligatures such as rope, belts, tape and cords, linked items such as envelopes and stamps, bedding such as sheets, blankets, comforters, pillowcases, pillows and mattress pads, cigarette butts or other smoking devices, condoms, drinking containers such as cups, cans and bottles, handled items such as weapons and tools, other household materials such as towels, used tissues, toilet paper and paper towels, biological reference standards from known individuals such as buccal swabs from a victim, suspect, etc. and secondary reference standards from missing persons such as toothbrush or hairbrush should be collected. Next, we will study about the evidence collection. The collection of evidence should be carried out in a careful manner to prevent any sort of contamination and degradation. Careful handling ensures integrity during all phases of investigation. The materials or tools used for the collection should be free from human DNA because DNA cannot be destroyed by all germicidal treatments. For example, alcohol and hydrogen peroxide does not destroy DNA. Cleaning of the collection equipments should be done by wiping it with the fresh 10% bleach solution of 10:1 water to bleach ratio. Since all the evidences are subjected to deterioration, careful collection and storage of the evidences is necessary. Presence of dirt, grease or any other contaminant can seriously compromise the DNA typing process. Such issues will not change DNA alleles from one to another, but it will affect the ability of the scientist to obtain complete DNA profile. Biological evidences can be transferred to an individual's body or clothing or to any other object or crime scene directly. Liquid specimens deposited on the surface and forms stains. Non-fluid biological evidence such as tissue, bone or hair can also be transferred by direct contact and by deposits. These specimens can be transferred to a victim, witness, suspect or any other object through any intermediate medium. In case of the secondary transfer, direct contact between the original source and target surface is not present. The biological evidence will get absorbed into the surface or will adhere to it. Liquid samples will get absorbed, whereas solid samples will adhere to the surface. The state and condition of the evidence is always kept in mind before application of appropriate method of collection is carried out. Now, we will study about the collection of blood samples. When collecting blood evidences, the investigating officer should establish the location of the evidence and the position of the article with respect to other stationary objects at the scene through copious notes, sketches and photographs. Note the direction, size and the number of stains. Note the condition of the stains, whether they are dry, sticky, wet, crumbling, puckering, contracting or brittle. Note whether the stains are contaminated or exposed to natural elements or heat. The facts should be mentioned in the forwarding letter to the expert. Write the method of collection, whether the stain was scrubbed, cut, lifted, dislodged or collected in the form in which it is being sent. Pack the blood stains from various sources separately in suitable bottles or cellophane envelopes of appropriate sizes. Preserve the purity of the stains. This is achieved by preventing mixing of the stains with one another or rubbing the stain with the container or with other parts of the article. Preserve the continuity of the chain of possession and distinct identity of the evidence through proper packing, labeling, sealing and observing the other legal formalities. The possession of the evidence should be accounted for from the time of recovery to the time of its production in the court. Method of collection of the blood evidences vary with the nature of the evidence and the place or the article on which it is found. Now we will study about the collection of liquid blood samples. The liquid blood is found at the scene in the form of blood pools. 2 samples, each of 5 milliliters, are collected in clean vials. In one, sodium fluoride, oxalate or citrate is added and the other is kept without any preservative. It is preferable to collect and send the liquid blood in sealed tubes without the preservatives. The tubes are cooled in crushed ice contained in a thermos bottle and sent as such to the expert. A third sample is collected on a piece of a clean rag. A stain on a piece of clean white cotton cloth about 3 cm square with some unstained part of the cloth for control test is sufficient. The stain is dried and packed in a clean envelope or bottle. Needless to mention that the materials should be uncontaminated and dry. If the blood in the pool has dried, about 3 grams of the dried blood is collected. If the blood in the pool is a sticky mass, a piece of cloth is stained in the center of the pool, dried and packed as usual along with the 3 grams of dried blood. Now, moving on to the collection of wet samples or wet stains. Sometimes wet stains are found on the scene of crime. The exact technique for the collection will depend upon the nature of the surface and the quantity of the blood. The following are the general guidelines that we will study here. A clean piece of rag or filter paper is taken and pressed against the stain. The stain is transferred to the rag. If the stain is partially dry, the piece is moistened preferably with saline water. It is pressed against the stain. The same is transferred onto the piece. A cloth piece is always preferred over to filter paper. If a stain is on the non-absorbent surface, it is allowed to dry and the article is collected. If the stain contains sufficient liquid blood, it is collected in the same way as the liquid blood. If the stain is on the absorbent surface and has penetrated the surface, it is allowed to dry and the article is collected. If it is not possible to collect the article, for example in case of the stains on earth or unpaved flooring, the stained cone of the material is collected in a glass jar. A controlled sample of the unstained material from the place near to that of the stained area is also collected. Now about the collection of dry stains. In collection of dry stains from the scene, the following situations are common. First, on clothes. Take possession of the clothes bearing the dry stains. The number, position and size of the stains are properly recorded. The stains on different clothes or on the same clothes are not allowed to contaminate one another through dislodgement during packing or transit. On the absorbent surfaces. The absorbent surfaces absorb the serum. Therefore, for correct sampling, stained and unstained materials are separately collected. On the non-absorbent surfaces. To collect blood from the non-absorbent surfaces, one should collect the article wherever possible. Moisten a white clean piece of cotton rag or filter paper with saline water and press it against the stain. The stain is transferred to the piece. Moisten a piece of rag with inert gum and stick it to the stain. The stain adheres strongly to the surface. Peel off the piece when it is dry. The stain is transferred to the piece. Dry it and pack it as usual. If there is no alternative possible, scrape off as much as the blood is possible with a clean razor blade on a paper from the stain and pack it in a clean envelope or vial. Ordinarily, scraping from blood stain is not recommended as it is likely to result in the loss of some blood. On grass, when the blood is found on the grass, one should tie the grass with a thread, cut the tuft, dry it and pack it in an envelope or glass jar. Take care that the blood does not get dislodged in the process. When found on the body, the blood stains are sometimes found on the body of the culprit at the scene of crime. They should be carefully handled to avoid contamination from the body secretions. If it is found in the hair, it is collected by combing the hair. If sufficient quantities are not obtained, the hair are cut and collected. If the blood is found in the fingernails, it is scraped out with a clean toothpick. Collect control sample from unstained fingers also. The paring nails is also not recommended. If blood is found on other parts of the body, a wet piece of clean rag or filter paper is pressed against the stain. The stain is transferred to the piece. If the stain is adhering strongly, the piece of the cloth may be moistened with inert gum solution. Then the washed bloods. If the blood found at the scene of crime or on the clothes has been washed, it is still possible for the laboratory staff to detect traces of blood from the washed materials. Next, about the blood samples. The evaluation of blood evidence is a difficult task. An experienced expert handles the work. The collection of blood samples from suspects and victims is necessary for reference. These samples consist of liquid blood. Qualified medical personnel should always collect blood. In case the person has received blood transfusion, then the forensic science laboratory should be informed. 2 tubes of 5 ml each should be collected in vacutainers with EDTA as the anticoagulant. In case of collecting post-mortem blood, it should be collected from the non-body cavity areas such as heart or major internal blood vessels. Next, we will study about the collection of semen stains. Visual location of semen is a difficult task because the stains are almost colorless and can be destroyed by washing. Semen stains are found on the persons of the victim and the culprit, on their garments at the scene of occurrence and in vehicle if one is used for the commission of the crime. Collection from victim. The victim carries semen stains on the affected parts, on the skin, hair, close to these parts and on the clothes, especially on the undergarments. All the clothes should be taken into position. The help of a doctor should be obtained to collect the swabs from the affected and close by parts of the body and the hair suspected to carry semen stains. Collection from culprit. Seminal stains may be found on the flies and front parts of trousers, undergarments and on the front or back flap of the shirt of the culprit. The culprit may have used handkerchief or loose piece of cloth, paper or fibers to wipe off the seminal fluid. It may be found in his pockets or at or around the scene of occurrence. The culprit's sexual organs and skin close to it may carry the seminal stains. Pubic hairs of the culprit are also likely to carry seminal stains. Next, about the collection from the scene of occurrence. Semen stains may have been discharged at the scene. It may be found on the floor, carpet, furniture, bathroom, lavatory, on the bed, on draperies, or on discarded pieces of clothes, garments, and paper lying at the scene or thrown in dustbins, waste heaps, or closets. If it is an outdoor scene of offense, the semen may be found on the surface, grass, leaves, discarded pieces of clothes or paper. The victim can help in locating the seminal stains or semen-stained articles. If the vehicle has been used for the commission of the offense or crime, it is likely to carry the seminal stains, including the article used for wiping off those seminal stains or fluid. Next, about the clean collection practices. Some of the ways to prevent the contamination and degradation of the biological evidences are: the disposable latex gloves should be used to handle the evidence. When field testing evidence, swab the stain and test the swab rather than directly testing the stain. If the stain is small, it should be tested in a lab rather than in the field. When it is necessary to dampen a swab to collect a dried stain, any source of water that does not contain human DNA is acceptable. Sterile water, distilled water, saline solution and tap water meets this definition. Document which type of water is being used. Dry damp items and swabs. When it is not possible to thoroughly dry the item, packaging such as paper bags will allow the drying process to continue. The wet items may be dried by hanging or by laying out on a clean surface indoors away from the scene of crime. Package each item separately. Other biological samples are also collected in the same manner. Next, we will study about the packaging of evidences. Various points should be considered while packaging the biological evidences at the crime scene. Package evidence and seal the container to protect it from loss, cross-transfer and contamination. Seal the package in such a manner that opening it causes obvious damage or alteration to the container or its seal. Use paper bags, envelopes, boxes and similar materials for all biological evidences. Avoid plastic packaging. Make sure that all swabs and evidences are dry. Package each item separately. Avoid mixing of items to prevent cross-contamination. Swabs collected from a single item may be packaged in the same container. Mark each package with a detailed description that includes the item, location, where it was collected, name of the person who collected it and the date of collection. Seal each package with tape. Staples are not considered a proper seal. All seals must be marked so as to identify the person making the seal. Mark through the seal with name or initials and date. The integrity of the item often is maintained through the package's documentation. The documentation includes all the markings, seals, tags and labels used by all the agencies that are involved. Therefore, it is critical to preserve or document all the packaging and the labels. Next is documenting evidences. Locating or location of the evidences at the crime scene should always be noted down. Photographs and placards should be used to document the location of each evidence. Description of the evidence should be done in detail along with the name of the person who collected it and the date of collection. Sketching of the scene should also be done to record the evidences. Next, we will study about the storage of evidences. Storing evidences during all phases of investigation and litigation is necessary. The evidence should be dried and stored. Storage should be in a facility that minimizes extreme heat and humidity which may lead to DNA damage. Dried and odorous evidences should be kept in a sealed plastic bag. Those evidences which cannot be dried should be frozen. Whole blood should be spotted onto a sterile cloth, bloodstain card paper or the FTA paper by the expert for storage at room temperature. Sealing of the packages is necessary in order to prevent the contamination and degradation of the evidences. Packages from the same case should be stored in the fewest number of containers using boxes or large bags with care taken to avoid contamination of the evidence. In case of large evidences such as cards, mattresses, etc., which cannot be stored for a long time, they should be photographed showing the location of the evidences. Now we will summarize all that we have studied in this module. The collection of evidence should be carried out in a careful manner so as to prevent any sort of contamination and degradation. Liquid specimens deposit on the surface and form the stains. This state and the condition of the evidence is always kept in mind before application of appropriate method of collection is employed. Visual location of semen is a difficult task because the stains are almost colorless and can be destroyed by washing. Disposable latex gloves should be used to handle the evidences. Use paper bags, envelopes, boxes and similar materials for all the biological evidence. Photographs and placards should be used to document the location of each evidence. Dried and odorous evidences should be kept in a sealed plastic bag.